Um, my name is Michelle Pagani, and I am showing you today how to use your Datahub Probeware with AP Bio investigations. I used to teach AP in the old curriculum, but I am familiar with the new curriculum as well. All right, so the objectives to, I'm sorry, can you go back one? There we go. Can you just make sure that your phone is muted by pressing *6? And then if you have a question at any time, you can unmute it with *6 again. And then if you have a question, you can chat it through the WebEx on the computer. All right? Any questions yet before I get started? No, we're good? All right. So the objective today, as you can see with PowerPoint, is teaming technology with wet lab experiences to the AP Bio investigations. Some of the investigations I'm going to talk about today aren't necessarily word for word College Board. I'm going to give you some alternatives as well, but most of them are going to be based on the College Board expectations that are in the lab manual. All right? So this is our, can we switch? This is our Datahub, if you can see it here, and it has 13 different functionalities on it, 13 different things that you can measure with it. Some of these buttons, there aren't 13 buttons obviously, so some of the buttons have dual and tri functionalities on it. They're really easy to use, and you can read it or control it by an iPad, which is the easiest way to do it. You can control it by this device so that you don't need to have anything else. You can take like a one measure or one reading at a time using this device. Or if you use an iPad or a computer, then you can actually trace more than one utility at the same time. So what I've done here is I've set it up for the first one. Go back a second. So this is on an iPad, and the way that you can get it onto an iPad, let's go to the next screen. Ways you can use the Datahub. We'll start with that. So it's data collection of the dependent variables. So in some cases you can actually measure what you want to measure. The second item is you can measure the independent variables, or what is the environment like at the time that you take the readings. And the third way is it gives you other ideas for how to stretch the inquiry section of the investigations. All right? So it's really easy to use. With an iPad, what you're going to do is you actually go right, as any other applications, you go to the store, the application store, iTunes, and you download the Ward's Datahub application, and it will give you everything you need that's listed here. And I'm going to draw out some other things that are in it as well as we go further down the presentation. The computer, you end up, when you buy the Datahub, it will actually come with a disk that will allow you to download the software, and you can then control it from the computer. It's easier on the iPad. I think most people have iPads, either personal or at school, and so, or they can rent some out for the day, lease one out from the IT section. So we're working on that today. It's a little easier than the computer. All right, so I've listed here the easy way that how things get started on the Datahub, and the information that comes with the Datahub has all the what button to push, what time to push, and it's not only just in words, but it's also in visuals, so little pictures, which I always find easier to follow sometimes. All right. So if you see here, it just says turn on your Datahub, and you launch the app, which I've already done here. Then you have to pair your iPad to the Datahub itself via Bluetooth. So if you have any Bluetooth device, it will actually connect with this. So you could even do it on phones if you wanted your students to access it with their phone instead. So these are always wirelessly transmitting. It makes it easy, no wires to trip over or any of that such. All right, so the first of these experiments that I want to talk about is transpiration. And I kind of skipped the beginning for now as far as labs 1 through 10. Some of them pair very easily with the Datahub. Other ones are a little harder, which I'm sure that you can understand that. Transformation, bacterial transformation, there's not much you can do with a Datahub other than maybe check the turbidity of the solution before you inoculate it onto the plates. So I started with ones that I think were indicative of or examples of either checking that dependent variable or checking those independent variables that I spoke about earlier. All right, so the first one is the lab 11 transpiration, and I've set up, you might want to switch, there we go. So I've set up the transpiration experiment here, and I think that this Datahub can best be used by tracking these items, and I've set up this to be on what I think goes with this experiment. I think you can see it pretty well, right? If you, along the side, there are buttons that kind of tell you how to do each of the things. We're going to talk about a few of them. The one in the middle that looks like the Datahub, very similar. See, it looks like the Datahub in the middle, and it has a little Bluetooth symbol next to it. So that's saying it's reading that Datahub. Then, and if you have multiple Datahubs, that's okay too. You only have one Datahub to one wireless device, but on the back of it, if you can see, it's got a number, and that number will actually come up in your iPad general settings to pair them. It goes through it very well in the booklet, or you can call us for help as well. So if I click on that button that I showed you before that looks like the Datahub, it gives me all of the different sensors that were listed that I didn't actually talk about yet. And if you look at them, the possibilities are air pressure, barometer, pH, dissolved oxygen, conductivity, light, external analog sensor, which then can connect with vernier probes if you want to combine the Datahub with a vernier probe, ambient temperature, thermocouple, pulse, external temperature, humidity, GPS, colorimeter, and turbidity. So today for this one, we're going to be testing the environment, so we're going to be testing the independent variables, and I've chosen barometer, light, ambient temperature, and humidity. All right, and so all I've done is go through and click each of those, and you can see they turn on and off really easily. And then you set how many, what's your rate going to be and how many samples per that rate. So we're going to say for every second that there is 100 samples that we're going to collect. That's what that's saying here. And since it's already paired, if we want to take how it appears right now, we can put this right next to the experiment. So it's right next to where the plant is in the potometer. And then we hit, I'll show you again. You see the little running man right there? Okay. And then it turns to a stop. See the stop? So if I hit the running man, it will start taking readings across the screen. You can see it continues to go across the screen. Also, I wanted to point out on the PowerPoint that you're able to see, there are some new drawings. We tried to make the literature more understandable, so you can see some of the, especially the figure 1 there, is one of the new illustrations that we've added. All right, so you can see that it's staying basically the same all across the screen, which you would expect it to stay the same since we haven't done anything to it yet. All right, so some of the ways that are suggested to change things for inquiry are to add light or add a fan. And what you can do is have different groups do different things. And you can put a light on your plant. Might have to get a little higher. And as you do that, if you expose the light portion of this to the, if you expose the light portion, it's going to actually start reading the increase in light so that they can get readings on what it was normally and once it's added. So if I hit run again, you can see the light is increasing, and as that light increases, it's also going to, as we know, increase the temperature because the light bulb is going to give off heat. But you can see differences in this one versus the last graph. All right. Now somebody wouldn't have to hold that. You could put it on some kind of another clamp off of this, but it does have to be exposed to whatever the plant is exposed to. All right. Then also the readings are not only just on the graph, but they're also on the readout on the Datahub. You could have one group testing the light. We could test a fan. We could test the sprayer to increase the humidity. If you're going to test the one where you put a bag around it, which is the normal way they've done it, you're going to have to put the Datahub into the bag because it wouldn't be a good example of, it just wouldn't be in the same environment as the plant is. So make sure that if you do that, you include this into the bag. Like I said, you could hold on to a clamp here and then just bag the whole thing. It should be pretty easy to do. And because this is Bluetooth, even if this is in the bag and you think it's hard to read, it's actually reading it on the iPad for you. All right, so I said that each of these have 2 or 3 functionalities, and if you look at this, let me show you what I meant by that. You can see it says pH, and it has an omega, I believe, upside down omega, which I'm trying to remember what that one is, and a DO2. So this one has an outlet next to it. If you go around, you can see that there's a plug in each of these. So each one of these have different plug-ins, and you can see that this is the plug here. So a pH meter will plug in there, a dissolved oxygen meter will plug in there, or a conductivity will plug it in there. And that's why I said that some of them have 2 and 3. So depending on which one it's saying on the little screen, the screen tells us what it is. This one says ambient temperature. If I want to change it, I can just click this, pick this one. This one says external temperature, and it says heart rate. So it actually does 2 different measurements. If they're on the same button, you can't really measure it at the same time. All right. So that's the transpiration. Are there any questions about how you might use transpiration with Datahub? No? Okay. So the next one is animal behavior, Lab 12. And with animal behavior, it's going to be, I wanted to show a couple of things. One, I brought out the tray that most people are used to seeing, which is the choice tray where you have 5 different sections. You would normally use this with pill bugs or sow bugs, but what we're going to focus on today is the Drosophila. And the Drosophila ones obviously aren't going to stay in this tray very well, even with the plastic lid. So you have to make a bottle out of 2 plastic bottles, right? Cut off the bottom, tape them, and you can have flies in here. Now, how can you do different choice chambers? You could do vinegar in one side and water in another and make it A and B and see how many flies go to one side or the other. Or if you want to do something that's more quantitative as far as experiential conditions, then you are going to, you could take some black paper, put it on one, take this light, turn it on so that it's on both parts of this, and then after a while, obviously the dark one is probably going to warm up a little bit more than the clear, and you can then measure inside the chamber before you add the flies. If you try to measure after you've added the flies, you're going to be catching the flies. So a good way to do this is to take your Datahub again, and now we're going to test something different. We're going to use an external temperature, and for the external temperature, unlike the variables we just did, you actually have to change, you have to actually add a probe. So if I look at this right now and I want to change what I'm going to measure, I'm going to go back to the screen that we did before. I know I'm off a little. Going back to the screen we did, we're going to turn off the ones that we just did, and we want to see what the external temperature is. So if we want to look at external temperature, we're going to take this and plug this into where external temperature goes. One way that you know how is that each of these different sensors, most of them have different ends so that you know you're putting the right one in. And wherever you are putting it is also across from wherever that condition is measured. So this is where the probe goes, and the button has a little thermometer on it. And so that tells me that I'm putting it in the right place. All right, so once I have it there and I have it measuring that, I can start, and I can take this external thermometer and I can measure inside where the black is, measure inside where the clear is, and then once I do that, I can take my flies and I can shake them into this bottle and then watch which way does it go, where it's warmer or where it's actually cooler. All right. All right, let's see. Oh, another one that I have listed here, you can test the light. If you want to try light on it, between it, you can try relative humidity. You could make one more damp than one side, more damp than the other. You can do the external temperature that I just talked about. You can see what the temperature, does it change for any of those variables when it's cooler or warmer. And the last one, sort of last one that I listed there was pH. And for pH, we always say that it is attracted by acid, or that's one of the things we test, and we always test vinegar. But vinegar is a very fragrant acid, so we might want to see if it's due to it being an acid, or is it due to it being that particular fragrance. So you can take a pH of about 2, and you can take some vinegar. And if you want to prove or have the students do this, they can take the pH using the pH probe. That's where pH comes in for this one. So they can take the pH probe and connect it to the Datahub. So I'm going to stop the temperature. I'm going to take the pH probe, put it into where the pH probe goes. And then I would have to change this to be pH, and I could measure it that way. Because the pH isn't changing much, what I suggest is instead of actually putting it on the iPad, this is one where you're taking one measurement. Because you're taking one measurement, you can do it up now to me. Thanks. Because you're doing one measurement, you can just take the pH probe and you can take that measurement directly off of this. You have to change it to pH. You can see a pH is there. Now you can do it down. Sorry, I know I'm switching back and forth. And it's going to take the pH. This is a pH buffer of 2. As you can see, the numbers are about 1.98. I think it came pretty close. So they can compare between that pH and the vinegar pH and see how close it is, and test the flies to see which they prefer, or the sow bugs, because it comes with the kit comes with both. All right, what we've added along with this, what we've added along with this now is College Board suggested that you also test not only the adult flies, but you actually test, there we go. If you can see, there's some larval forms. So you can do the same sort of experiment on the adult flies as you do the larvae, or maggots if you really want to say it. You can put acid on one side, water on the other, and you can actually even take and carve out a little bit of the agar if you want it to stay in one spot. Then you put the larvae in the middle, and you see which side does it go toward. These have really made some nice patterns of, don't know if you can see it, but it's kind of, if you can see that nice little patterns all over the agar there. When you're done, you're just going to bleach these like you do any bacteria, because these little guys have a lot of bacteria on them, and then you can just toss them like anything else. All right, so this was actually recommended by College Board, and we just added this. So if you order this kit and you get nutrient agar plates, and you wonder, why do I have nutrient agar plates with this kit? That's what it's all about. All right, next slide, please. So the experiment I just talked about was what's listed there at the beginning. The new experiment added aligned with the College Board. And the question that you can pose, or they can pose, is do the larval forms have the same attraction as the adults? So if the adults are attracted more for the vinegar or for either acid, are the larval forms the same? I think probably, because they're both going to be food sources for that, for those animals. But, oh, and I also need to add, when you do, if you have them do the larval forms, make sure that they don't take third instar. If they're starting to pupate, they're not going to go across the agar one to the other. What they're going to do is they're going to go up the agar and try to get out, because part of the pupation is to get high up so they can hang as cocoons. So just as a side point there. So the other thing that was interesting that I recently found is on the rest of the slide it says, can we test the following article's findings? It says that fruit flies, they flee from shadows, and that was in the Science News just last week, 2 weeks ago, from June 13th. And they found that flies, even if they were hungry and they were being fed, they would get afraid of shadows going over their enclosure. And so they would go and stay away from those shadows, and they would get so traumatized, so to speak, that they wouldn't even go by their food for a good, say, 20 minutes. They likened it to a gunshot for us. If we heard a gunshot, we'd stay away. Same thing for them. They are proposing in the article that the shadow is actually reminiscent for them of some predator coming to eat it. So anyway, it's interesting, and it's something that you can add to say, hey, these are new findings. You think that this is just an experiment that the College Board is telling us to do, but actually people are researching it every day in real life, because sometimes they don't think that it correlates well. All right. The last of the three that I'm going to show you is the enzyme catalysis. And the enzyme catalysis, or energy dynamics, I think, well, enzyme catalysis is when you take the lovely smelling glycol. It smells to me similar to fly nap. Some people complain about the smell of it. And you mix it with peroxidase from a turnip, and it will turn a color. So you can see this is the one that has some color, and this is the one that's my blank. It just has water in it at this point. And what I've done is made a somewhat color palette of varying concentrations of the enzyme substrate, just like you would in the experiment. And in there it talks about a colorimeter or a spectrophotometer. If you don't have a spectrophotometer available, what you can use is the Datahub. And with the Datahub, what you're going to do is, instead of doing it on, you don't have to do it on the iPad on this one, you are going to use it as a single unit. And on here, there is something that looks like a cuvette with arrows going through it. This can serve a few purposes, actually. It's a colorimeter, and it's also a, I don't think turbidometer is a word, but it tests turbidity, and it also tests 3 different wavelengths of color. So it doesn't say, you can't set it as you normally see to 680 or 740 or something on a spectrophotometer, but it will give you a difference in readings. What you can do with these is you turn the Datahub again so that now you can see that there's an opening there, and that opening fits these cuvettes perfectly. These cuvettes are made for that. The cuvettes have a little top on them so there isn't any spillage into the device, because the device doesn't do well with liquids. So once it's capped and you put it into there, you can click that and you can actually see, you see there's a light. And so you can test turbidity, or you can test, in this case, it's the red, green, blue wavelengths, and then what is the absorbance, so how turbid is it? And you know that the absorbance is inverse of the transmittance, so it will, this number will change. So each one of these, if I compare this one, about 270, with the brown one, which has the enzyme substrate, you can see that it's almost 1450, which makes sense because the transmittance, this is not transmitting as much because it's darker. So you can use a colorimeter, the ambient temperature, so what is it like on the outside, and you can test the pH, because some of the things is testing the enzyme for different pH buffers. You can test the pH and have them work with different pHs for the inquiry. And you go to the next slide. Thank you. So I haven't set up all of the experiments, but I want to talk a little bit about how to use this in some of the other experiments that I haven't set up. One of those is the photosynthesis. What's cool with the photosynthesis one is you can use relative humidity, light, ambient temperatures, and I'm going to skip over the next one, the air pressure, for a second, and go to the colorimeter. Colorimeter, you're going to do the same thing we just did with the enzyme one, except you could make an extract of red leaf cabbage versus green leaf cabbage and see what the difference is on the blue, the red, and the green wavelengths. I tested this with blue food coloring just for fun, and the only one that was really different between blue and clear in a big way was the red, which makes sense because the red is going to read the blue differently. The blue is not necessarily going to be picked up with the blue wavelength because it reflects. So, all right, and the last one that's listed there is the air pressure sensor. And the air pressure sensor, there is, I told you that we were going to have some that aren't completely like the College Board. And this is an example of that. And I said that there were controls over here. There's two controls that I'm going to talk about now. One is down here, you see a little library, a little book. And what's cool about this, once you download the app in the iPad, it gives you access to a variety of activities that are already there. The only one that's really pertinent to biology, because it's a combination of all of them, is what is photosynthesis. This is the teacher's guide, and you can use this as an extra activity to go along with the Datahub. It has vocab. It's just like any other of the literature. And here's those visual cues that I said were in the instructions to make it easy. And in this case, it's doing it a little different. As you can see, it has the gas pressure into a syringe, and you can measure how much photosynthesis occurs there because of the pressure difference. And you can use that also in cellular respiration to measure it more quantitatively. The gas pressure sensor comes with the Datahub, and it looks like this. It's a little plastic tube with an adapter on it that connects to the Datahub. Let's say, and I know this never happens, let's say you have a student that's ill and misses a day, right? What you're going to do for this one is, if you go up to the first one, let me show you the first one, see how it's a little file here? I can save experiments. I've saved a couple of crazy ones there that were just practice, but it also has ones that someone else has already done the data, and look, there's photosynthesis, and it gives you data that they can actually work with when they've come back to school. Or if you want to send them the data, they're ill for a long period of time, or homeschooled, or lots of different reasons like that, you can send them a snapshot of this, and they can work the experiment out on their own. Okay, next, so that's photosynthesis. Cellular respiration. With cellular respiration, you have the tubes with the P's in them, or you can put, you can have a syringe with flies or pill bugs, or if you're really daring, you could do it with mice, right? With putting a mouse in a tube and testing it that way. You want to test the outside and compare it to the inside of the tube, because you, what you're usually looking for is a gas, oxygen decrease. And so that oxygen decrease is going to change the pressure inside that tube, and that's how you usually measure it. So it's good to know what the barometric pressure is outside from one class to the other in case you get different results. You can compare one class, say in the morning it's hotter and the barometric pressure is just a little different than later in the day, or there's a storm that moves through and changes things completely. You can document that and then compare it between classes. You can also, like I said, use this air pressure sensor to combine and test the cellular respiration that way. All right, next, mitosis and meiosis. One of the parts of the mitosis and meiosis is to grow garlic, and the garlic, you're checking to see what the mitotic rate in that root is. You might want to try adding things to it, say caffeine or acid, and see if it stunts the growth, if it helps it grow faster. And so you can use the Datahub to more clearly define those conditions. And next, energy dynamics. Energy dynamics is the experiment very much like artificial selection, which is lab 1, where you're growing plants. and you're comparing whether in lab 1 you're comparing selective artificial selection, and in energy dynamics you're actually checking to see how many plants are produced, and you're also testing how the butterflies eat the food and making the connection. So there again, it's over a long period of time. You might want to have them record what's the temperature every day versus what they eat. Do they eat more when it's hot? Do they eat more when it's, cooler? Along those lines. And here's a list of all the products that are in our line. And it also has the refills that we've just started offering, refills for a class of 8 groups. And so I thought I'd put it in a nice, easy format to read. It also lists where you could get more explanation about the Datahub, and there's some videos, and it's somebody other than me. So anyway, all right. I thank you for coming. If you want the PowerPoint, you can email me at the email that's on the screen, or you can contact Science Help, or if you ever have any questions about AP Bio or anything with bio, feel free to contact me. Thank you.